반갑습니다.
Good morning. My name is Kang Byeong-chul from Insilicogen, and I would like to thank the organizer for inviting me. And today, my topic is about genomics and big data from marine resources. And I will give you some case studies, and I will talk about how we can strengthen this capacity for the future. And the ocean is home to various forms of lives. So the bacteria made oxygen, and it was the beginning of the diverse lives in the ocean. And recently we got to know about the dark oxygen, and the ocean could be the first place where the different forms of lives were born. So as you see, the ocean is the origin of lives. So that is why these living species struggle with one another in the ocean, and in the process we develop our own defense mechanism. And out of the defense mechanism, we use diverse microorganisms and living species surrounding us, and through the interaction we further develop our own defense mechanism. And we focused on peptides out of diverse defense mechanisms. And peptide is a short protein, and by using this protein we can find diverse mechanisms, and there are about 1,000 known proteins, known peptides, and we have antimicrobial and antibacterial peptides and proteins which are fighting against other living species. And there are about 1,000 known peptides, as I said. And what we focused on was AMP, which stands for antimicrobial peptide. And I will give you some cases, and I will give you some potential for expansion. And peptide has shown its potential for different markets. And from 2010, there has been various products being sold on the market. And as you know, NVIDIA is an infrastructure provider, and they are now studying peptide as well. And this is the number of approval for peptide products, and there is an increasing number. It shows that peptide is stable, and it has various applications in the biological field. So we see a huge scalability of the peptide in the market. And I mentioned AMP, which is antimicrobial peptide. And there are two processes of antimicrobial activities. So we can directly target the microorganism, or we can trigger the immune system of the host so that the host could respond. So we looked at the direct process. That was our focus. And a small peptide has the characteristic of better affinity to bacteria cells. And with aggregation, they can destroy the cell of microorganisms. That's the process. So I will give you the details. And this is originated from a longer protein, but it was fragmented into shorter proteins, which generates various secondary structures. So this shorter protein is secreted outside the microorganism, and it can work as a peptide from them. And to screen peptide, we have to develop the library, and that is the general way. However, now we have a much more efficient way to do that. And one of that is multi-omics data. So from the genomics, we look at how it interacts with other living species and the surrounding environment. So we can assume the pasts, the latent pasts of the, and by using the pasts through artificial intelligence, we can predict and discover the new competence of the peptide. And we have 3 steps to do that. First, we identify the biochemical activity. And the second step is the application of AI. And the last step is how the peptide is interacting with the protein. So we can find out which protein it is interacting with through molecular docking. And let us talk about the first step. So as you know, proteins are already defined in the biochemical manner. So there are a lot of theoretical basis for these proteins, and the sequence is fragmented theoretically, and based on the theories we can perform the calculations. And we can develop this kind of large-scale metrics based on calculations, and there we can find the data which could be used for artificial intelligence. And for example, if we study the abalone, there are diverse proteins within the abalone. But before going through the process, we can look at the existing knowledge of other marine lives, and we have the information on them. That's why we can perform calculation faster, even for the new protein. And by using artificial intelligence, we can select a certain part of the marine life, and we can use different technologies. And we used about 15 AI models, and we selected high-performing models, and we have been optimizing them. And let me give you details on the models. So even though I say artificial intelligence, it is more of machine learning and deep learning. And these days we are utilizing machine learning actively because the biologically significant characteristics can be extracted, and we need to secure the explainability of them. So the data we extract should be applied to the medical field and the agricultural field. And to explain the phenomenon and to predict what would happen, we need to secure explainability beforehand. That is why we are using machine learning. And there is a difference from the existing statistics and the new AI statistics. And the existing statistics is looking at the linear relationship. However, AI could even give an answer for non-linear interactions. So we have different algorithms, but we focused on 15 AI models, and we evaluated all of them, but now we are focused on 5 highest performing models. And we built the data of 200 characteristics. And from these 200 data points, we went through all the learning processes so that we can examine the biochemical features. And this is the result. So you can see each characteristic and its correlation with our target. So you can see the mixture, so it is not always working well. So by doing so, I believe that we can get the help of AI. So we finished the examination, and I told you that we are focusing on 5 highest performing models. And now we have secured approximately 90% of accuracy. So this is the progress we have achieved so far. So for peptide X, if we find out it has the antimicrobial capacity, we have to look at which protein it interacts with through simulation. So we can do the simulation to examine that. So for example, here we have KAP1. So NRF2 is the protein it is binding with. However, if there is an external stress of oxidation, NRF2 is released and it will go into the nucleate so that it can trigger the defense mechanism. So our peptide could be binded instead of NRF. So NRF will be abundant and it could go into the nucleate more, and the antimicrobial capacity will go up accordingly. And this is the conditions for target selection. And we looked at the dynamics of the molecular docking. And we selected peptide X, and this is the result of our research. We looked at how much capacity it has with the human body and how much protein could be produced. So this is the examination table for a certain peptide, and we can explain the structure of the peptide as a result. And so if we examine about 10 peptides, we can find a high efficiency for 6 peptides in general. So these are the case studies which were conducted successfully, and it is not only applied to the marine bio field, but also to other sectors. So artificial intelligence and big data can play a certain role in marine biotechnology. So AI and big data can identify unknown information. So we can identify the traces of different marine lives, and we can find information about the peptide, which can be delivered to the human body. And AI could play the role. And by doing so, marine-derived peptide could be commercialized, and we can secure the original sources in the future. But to achieve this goal, AI and big data should be converged, especially for the marine bio field. Diverse marine resources, they are less known compared to the land species. So we need further information on them. And to find more information, we need to use AI to find more genomic data and information. Then at the end of the day, we can identify new biomaterials. Thank you very much.